I try to share my screen. I don't know if it happened or is okay. Can you see my screen?
Yep.
Okay. So then, first of all, I would like to thank you very much for the invitation, which was I very happy to accept because I had a great memories about Kyiv and the Lutsenko University a couple of years ago. We have been there with a couple of my colleagues from the University of Sport Science in Hungary. And so I was very pleased what I have seen there, the activity and all the young people who wanted to do research. And so today I will talk about the role of novel local mechanisms in the adaptation of coronary circulation to exercise. And I think it's important that to know that exercise elicits improvement over parts of the cardiovascular system. And because of that, I'm sure. And not just the cardiovascular system, but as we heard, that the nervous system, the brain function. And we also know that sedentary lifestyle is a major but modified risk factor for cardiovascular diseases. And we also know that even short-term exercise, a couple of weeks of exercise, improves the ability of coronary circulation to provide higher blood flow to the working cardiac muscle. Moreover, it has been shown also that moderate daily exercise, anything, just a couple of miles, is really improves hypertension or the lipid profile and also diabetes. So, and that reduces the incidence of coronary heart disease. Therefore, we were very much interested to learn more about the mechanisms which improves coronary circulation. And especially we are thinking about the local mechanisms, which probably was less known so far. And we know that neural, hormonal, and many other mechanisms regulate coronary circulation, but we focus our study on the local hemodynamic factors which actually affect the coronary vessels, such as the pressure, the intraluminar pressure, the wall-shear stress, which affect on the surface of the endothelium inside the vessels, itself the flow as a mass transport, and also I will talk about the crosstalk between blood and cardiac muscle and oxygen consumption. Now, one of the very unique feature of the coronary blood flow that it is phasic. That means that there is a great changes in the pressure and also in the flow. And as we see in the left coronary artery, the flow is actually very low during systole or even negative, but very high during diastole. So both the pressure and the flow changes, from time to time. And if you illustrate this, there is a pressure which acts on the vascular wall tangentially, and there is the wall-shear stress, which is along the vascular wall inside. And here you can see a nice picture of Steve Siegel, and he stained the smooth muscle in the coronary vessels. You see these red dots, which is going, it's a circular muscle around the vessels, and you see the endothelium, which is stained in green. So the pressure mostly affects the smooth muscle, and the wall-shear stress affect mostly the endothelium, as I will now show to you. So these forces constantly affect the smooth muscle and the endothelium, and then elicit changes and elicit function, and there are so-called mechanotransduction, which then change the hemodynamic forces to diameter changes. Now, one of the characteristics of coronary circulation is that the oxygen extraction is very high, even at rest. So therefore, if you want to increase the oxygen supply, you have to increase by increasing in flow. So therefore, the increasing in flow has a special role in the coronary circulation. And then there is a rapid changes in the diameter, during the cardiac cycle, which is necessary to maintain the balance between metabolic demand and flow supply. It is just an illustration how great changes can occur in a small arterioles, can increase the size double the diameter. So there's a lot of room for increase or decrease in blood flow in the coronary circulation. Now, in order to study the local mechanisms, we isolated the vessels from the heart. As you can see here, it's a small arteriole, maybe 100, 120 micrometer in diameter, and a human hair is about 100 micrometer in diameter, so those are very small so-called resistance vessels. And then changing their diameter really influence the changes in blood flow. And because we isolated these vessels, the neural or humoral or other extrinsic factors were excluded. So we could only investigate the wall of the vessels, what happens when we change the pressure or change the flow in an isolated situation, and we use physiological salt solution instead of blood. So then first we started to investigate the effect of intraluminal pressure on the diameter of the isolated coronary arterioles. And then we also had the rats run a treadmill for 4 weeks with increasing intensity. And usually most of them are really like to run. And if you just put a wheel in their cage, they also run. So they like to run. So we didn't need to force them. And here is the experimental setup. We have the isolated arterioles here, and then we could control the pressure, which goes in and goes out. Thereby, we could elevate both sides and then elevated the pressure, or we elevated one and reduced the other side of the pressure, and there was a pressure drop, which then elicited the flow. As I said, the diameter about 119 micrometer. And then we could follow the diameter with a television microscope system. And here you can see, it's a little bit busy slide, but I will tell you that you can see the intraluminal pressure we set up, and then you can see the internal diameter of the vessels. Now, these two upper line, the red and the black, is when we take out the calcium from the solution. So the vessel passively dilate as a function of intraluminal pressure. Once we have the calcium in the solution, the vessels constrict when the pressure goes up. This is a well-known myogenic response, which has been described in 1902 by Bayliss. Now, if we exercise the rats, then we got to the green line, indicating that the pressure-induced constriction greatly increased as a result of exercise. Now, if we use an inhibitor which inhibit the nitric oxide synthase in the endothelium, you can see that purple dots when sedentary vessels, and the blue one on the exercise vessels, indicating there is a much greater nitric oxide influence on the myogenic response in exercised animals. Then we studied also the effect of indomethacin, a well-known inhibitor of cyclooxygenase 1 and 2, which is producing the prostaglandins. And as you can see that in the sedentary rats, there is a little dilation to indomethacin, indicating that there is a constrictor prostaglandin is released in the sedentary, which is not there in the exercise rats. So in conclusion, in sedentary animals, in the coronary vessels, there's little modulation by nitric oxide and larger modulation by the constrictor prostaglandin. Exercise increase the myogenic response, increase the modulation by the dilator nitric oxide, and reduce the modulation by the constrictor prostanoid. And what is the physiological importance of these changes? That is that when you have the coronary perfusion pressure, and there is a so-called autoregulation, which means that the flow is somewhat independent of the pressure, this range is extended by this enhanced myogenic response to the lower level and to the high level. So at the lower pressure, you still can produce a blood flow, and the high level, you can also maintain blood flow. So you extend the range of autoregulation as a result of exercise. Now let's see what happens when we increase the flow, shear stress in this isolated vessel. Here you can see the illustration of the shear stress. This is the endothelium, and this is the blood flow velocity. There is a velocity profile, and then this has a shearing effect on the endothelium depending on the velocity and diameter ratio, which is the force shear rate, and the U is the viscosity. And when you have this one, that's the wall shear stress, the tangential force which exerted on the endothelium, which is sense the wall shear stress. Now, this is fairly new because for many years it was not appreciated. This force only the pressure. But then they realized that shear stress is also very important. How does it look like? If you increase the inflow pressure in these isolated vessels and reduce the outflow pressure, then there is a pressure drop via the vessels, which then elicit flow. And as you can see, as the flow increases, the diameter greatly increases. And this is due to the increased shear stress because the pressure here is kept constant. Here you can see on a summary slide that increasing in flow elicits a very substantial increase in the diameter, which is, of course, eliminated once we remove the endothelium. So it's mediated by the endothelium, sensing more shear stress mediated by the endothelium, as opposed to the pressure, which is mediated by the smooth muscle. Let's see the signaling of the wall shear stress-induced dilation. Here you can see that when we increase flow, and here you see the calcium signal in the endothelium, it's elevated a little bit, the calcium signal. But if you give acetylcholine, this elevation is much greater. Why acetylcholine? Because this is a kind of gold standard for the endothelium-dependent dilation, which was discovered by Furchgott and Zawadzki in 1980s. And basically, since that, we are aware of the fact that the inner surface of the vessels, the endothelium, is very important, both in health and both in disease. And when you give acetylcholine, the calcium in the endothelium greatly increases, but shear stress increases only a little bit. And here is the summary slide. You can see acetylcholine elicits a great increase in calcium in the endothelium, and shear stress a modest increase in calcium. Why is it important? Later on I will tell you. But here you can see another factor, the hydrogen peroxide, which is done by Gatterman, a group, that if you increase flow 2 minutes, 5 minutes, 30 minutes, you see an increased green staining, which is indicating the presence of hydrogen peroxide, because this dye is sensitive to the level of hydrogen peroxide. It means that also flow elicits an increase in hydrogen peroxide. And if we would summarize this, then you can see that wall shear stress, when increases, activates cyclooxygenase, which then elicits prostaglandin I2 and dilation. Also, endothelial nitric oxide, and that releases nitric oxide, also elicit dilation. Also, other factors such as EDHF, hyperpolarizing factor, hydrogen peroxide, and perhaps other also release depending on the region, like in kidney or in mesentery or in coronary, that could be different. And then here what happens is that really increasing cyclic GMP, and then which is decreasing the calcium level or increasing the calcium sensitivity of smooth muscle, and that's the reason why there is a relaxation of the smooth muscle. Now let's see how the regular exercise affects the shear stress dilator mechanisms. Again, here we had the rats running on the treadmill and then isolated the coronary arterioles. Here you can see the effect of flow on the diameter in sedentary animal. You can see a nice dilation as a function of flow. However, if we took out the vessels from the exerciser, this dilation was much, much greater, indicating an upregulation of the sensitivity of shear stress of these coronary vessels. Here is the summary data. This is the dilation to flow of the sedentary vessels, and this is the vessels which were isolated, coronary vessels isolated from exercise rats. Again, what we have seen that what is upregulated, both the nitric oxide and the prostaglandin part is upregulated as a function of exercise. Why is it important in general that when you have an increased dilation, when the shear stress increases? Here is a control situation when there is an increased pressure drop via the vascular network. The pressure drop could be due to the increased systolic pressure, and then venous pump or dilates or metabolized on the very small vessels. So the blood flow velocity increases. And when the blood flow velocity increases in a fixed diameter, that increases shear stress. And then when shear stress increases, the whole networks dilate. So that's important because you can have a much greater increase in blood flow with a much less work. So the heart doesn't need to work that much hard in order to provide a great blood flow to the tissues. And it's in exercise very important. And if you teach your vascular system to dilate very fast, when flow goes up, shear stress goes up, then your heart rate doesn't go up that high, your pressure doesn't go up that high. So you can really see right away who is the exercised or trained person or who is not. And in part, this is due to these shear stress mechanisms. And here is another illustration of this, that when we calculate shear stress in exercise and sedentary rats and the change in diameter, you can see that when shear stress increases, the change in diameter maintains shear stress around 20 dynes per square centimeter. And if there is no, in a sedentary, when the vessels are less sensitive, the shear stress goes up to 30 or even higher, indicating that the circulation or the heart needs to work much less here than here. Sorry, because the energy you lose the energy when the shear stress is high, because that's where you generate here, and also you lose energy. Now, when you have a lower shear stress in the system, the energy loss is much less. And this is the situation when there is no endothelium, shear stress goes up very high.
Dear AKOS,
yes.
Will you cope with your presentation with 3 minutes?
Excuse me?
Will you cope with your presentation in 4 minutes?
3.
3. Perfect.
Okay.
Thank you.
Yeah. So as I said, therefore, the shear stress mechanism is very important. And here you can see the functional importance. The red one is the exercise. Coronary blood flow can increase due to the increased shear stress dilation and decrease due to increased myogenic constriction. There is a greater range here. So another interesting finding was a couple of years ago, maybe 20 years ago, that people find that endothelial nitric oxide synthase also expressed in the capillaries. Why is it there? Capillaries cannot dilate. But very importantly, here are the capillaries, capillary endothelium, and the nitric oxide synthase inside, and here are the mitochondria of the cardiac muscle. So not only oxygen, but nitric oxide can also diffuse to mitochondria. And then what happens that when nitric oxide and oxygen diffuses to the mitochondria, it affects the function of the mitochondria, because both oxygen and nitric oxide compete for the binding side of cytochrome oxidase. So in that sense, nitric oxide can affect the oxygen consumption in various conditions. And here is another example. And these are dog heart tissues. And Dr. Hinchey and group, they added bradykinin to this and then measured the oxygen consumption. Bradykinin releases nitric oxide and reduces oxygen consumption, which could be eliminated by inhibition of nitric oxide synthase. So basically in exercise, more nitric oxide is released and affecting the mitochondria, which then affect the oxygen consumption and also the substrate selections. So another one I would like to just touch upon is that when there is a blood flow, the hemoglobin binds also nitric oxide, not only oxygen. So when there is a release of nitric oxide from the endothelium, hemoglobin catch the NO and deliver the NO to a remote area which may be ischemic, therefore dilating there and bringing then oxygen. So in conclusion, regular exercise elicits functional and later morphological remodeling of the coronary vessels, enabling to increase the range of autoregulation and the maximum value of coronary blood flow. Mechanosensitive mechanism via release of nitric oxide, prostaglandin, and oxidative species contribute to the optimization of coronary microvascular network. The interplay among factors of blood, endothelium, smooth muscle, and cardiac muscle result in a more optimal blood supply, oxygen consumption, and substrate selection during exercise, supporting the increased contractile function of the heart. And I would like to thank you for your attention. And this is a nice book I received from you when I visited you in Kyiv a couple of years ago. Thank you.